Reproducibility of dipyotetic systems found in the literature is difficult when water content has been disregarded. This method provides a standardization protocol for the development of these systems. This technique will allow the reproducibility of dipyotetic system preparation, contributing to coherent results among the scientific community.
Dipyotetic systems have proven applications in many different areas, including therapeutics and biomedical engineering, biocatalysis, extraction, and CO2 capture.
Through a visual demonstration, we highlight the variations encountered in the different methods reported in the literature for dipyotetic systems preparation. This is often not reported in conventional manuscripts.
Demonstrating the procedure will be Rita Gameiro, a postdoctoral fellow of my laboratory.
To prepare natural DES by freeze-drying, first add 2 grams of citric acid monohydrate and 0.9530 grams of glucose monohydrate in separate containers. Add 10 milliliters of deionized water to each container and stir until the compounds are completely dissolved. Mix the 2 solutions together and ensure the homogenization of the final solution. Place the solution in a round bottom flask. Freeze the sample using liquid nitrogen. Place the flask in a freeze dryer for 48 hours to ensure that all